Dean of Postgraduate Research for the Faculty of Science and Agriculture and Engineering. And I thought I'd start off with this slide because it's 150 years anniversary this year from the creation of the faculty. And during that time, we've done an awful lot and we've made lots of changes, I think, both to the city and to the university and to the world at large. It's my real honour today to present, and get the slides in the right order in a few minutes. It's my real pleasure to introduce the talks today. And I'm particularly pleased to be doing so because despite the history of 150 years, this also feels like a bit of a new beginning. This is the first of the series of insights that has been in person for a long time, and this is the first of the New Voices series since the pandemic. So it's really a pleasure to be doing this anew, and to say, particularly as this is also a lecture about new voices on science, agriculture and engineering. All of the people talking tonight are postgraduate students, and postgraduate research students are really a critical part of Newcastle University and more widely of the research base in the UK. They contribute towards the development of knowledge in our society, which is really a key part of the contribution that universities make to society. People do PhDs for a very, very large number of different reasons, but despite this, many of them are earlier in their careers, and they'll be using their technical abilities to help transform our countries and our societies and our lives in general for the better for many years into the future. So tonight we've got three groups of speakers who are doing research which has got this capacity for transformational change. The speakers are, in some order on the pictures behind me, we've got Jess McCoy, whose talk will be on putting the dead to work, and then Chloe Grant is talking about inflammation, the cause of neuron reduction in hens with broken keel bones. And finally, the sugar team, who are Anna Chiara Scalzoni, Corin Murphy, Tom Waring, and Alex Stokes, who are studying the uptake of glucose by yeast during a zero-G flight. There'll be a lot of opportunity at the end to ask questions after we've had all three presentations, so we'll do all those together. And I'm looking forward very much to both the talks and to the discussion that they will bring. So if we could welcome our first speaker, who is Chloe Grant. Oops. Got them in the wrong order. Sorry. It's not my fault. Here we are. You need your clicker.
Hello, can you hear us all right? Is that okay? So hello, welcome to my presentation, where today we're going to talk a little bit about how we can put the dead to work. And we're going to look back and see what these fossils and all these dead things can tell us a little bit more about climate change and for the future. So I'm a paleontologist, and thanks to media such as Jurassic Park, lots of things like that, paleontology is really well known for looking at dinosaurs. But I'm a little bit more interested in what else is going on. So you can see in this picture here, we do have a dinosaur, but I want to look at the sort of past environment that this dinosaur used to like to live in. You see that on this side, we've got some ferns. Now these ferns possibly have quite a tropical element to them. So these could tell us a little bit more that perhaps this dinosaur really liked to live in more humid conditions, a bit warmer, a bit wetter than present day. You've also got this tree over here, which looks quite similar to your present day North American hardwood trees. And we know that those trees in the present really like wetland environments, very swampy sort of picture that we're starting to build up here. So you can see, even in the background, you can see that we've got some coverage from the trees as well. We haven't got that much herbaceous taxa on the bottom as well, but we're already starting to build up a picture of what sort of environment this dinosaur used to quite like. Maybe it's quite swampy, quite humid, a bit more than today. And we've done that just by looking at the plants, which are in the background of this picture. So my PhD is very much focused on looking at deep time. So in particular, I look at a time period from about 28.1 to 15.8 million years ago. You can see just over there as well. So I focus, my studies focus very much on the UK as well. And you can see I've got some study sites just up there, some of the study sites which I like to look at. So these study sites are really important because they have and contain sediment, which contains the fossils which I'm interested in looking in, which are the age that I need, which you can see from there. Cool. Right, together, we're just going to quickly try and build, I'm going to take you through how we actually build up these past environments. So what I'll do is I'll do some research before I look into this, see where there is sediment, that is the age I need, and I'll go out into a field, I'll dig it up, and get something quite similar to the core, which you can see here. And this is just for one section of the core that we're going to try and work through. So I'm about to show you some pictures of fossils which we look at. The fossils I look at in particular are very, very small, and the pictures I'm about to show you are fossils which are about 13 million years old-ish. So this is a picture of pollen, and it's from the pine family as well. And pollen is very specific because each of the species have very, very specific structures. So some of them could have pores, some of them could have apertures. They've all got the very own defining characteristics as well. But this one in particular, I know is pine because it has a main body, and it's got two sacs attached to it. These sacs are like wings, and this is literally how the conifer pollen flies really long distances. So already we're starting to build up that perhaps this past environment that we're looking at, there's very much, you've got a pine tree either far from the site or near to the site. But the story doesn't end there. We can still keep looking, see what else there is. This is one of my favourite pollen grains. It's from the Cupraceae family. Just because it looks like Pac-Man, it's very easy to recognise. The Cupraceae family has a very distinguished characteristic as well. But it's really different to the other one that we've got because you can see it's more perforated. It's got a different shape entirely, but they're both conifers as well. But the Cupraceae family, we know from looking at the present-day Cupraceaes, these ones very much prefer swampy environments as well. These are like the trees that you get around swamps. So already we're starting to build up that picture just by looking at what we've got here. But it's not just pollen that we can look at. We can also look at fungi. So I love fungi, personally. I think they're dead interesting because you can see a lot more about interactions between plants. Some of them can be quite parasitic. Some of them can have relationships with plants that benefit both parties as well. It's really interesting. That's just me. But this fungus in particular, we know it was discovered by my supervisor back in 2019. And we know that it prefers lake environment, like lacustrine environments that are very slow moving, quite shallow. And it has affinities with, if there's lots of rot going on as well. So already we're starting to build up that picture. So just from this part of this core here, we've possibly got a pine tree far away or close to the site. We could even have this Cupraceae where we've got some swampy sort of trees around, possibly a main swamp. And we've also got probably some dead trees in the background which have been eaten away by the fungi. But you're probably wondering, well, what is the point in doing this? That's fantastic, but it doesn't matter to me. Well, we use the principle of uniformitarianism, which basically suggests that the key to understand past processes is the key to understanding present processes, and vice versa. And we'll extrapolate that to future processes too. So this is my partner, Harley, here on the right. He's actually sitting in the crowd over there. And he runs global circulation models. And basically, he is modelling the future of climate change using what we already know from past and present climates. What's really important to notice as well, though, you need to have comparable environments in order to do this, which we're going to talk a little bit more about doing that soon. But you probably think, well, that doesn't matter to me. Climate change isn't going to affect me. That's for my great-great-great-grandkids to worry about. Not the case. So this is a picture taken from the Australian wildfires back in about 2019, 2020. And the thing is, with Australia, wildfires are quite common, as it is. So it's very dry, it's very hot, and also in the background of the picture here, you can see that we've also got some pine trees as well. And these have a natural fire regime. They're very dry, often get set on fire as well. But the thing is, nothing had been seen to this scale for a really long time. And it took the firefighters a long, long time to fight this and put it out. Lots of animals were displaced from their habitats. Lots of people lost their homes. There was a massive upheaval about it. But you're probably thinking, well, we all know about climate change. Why living in Australia then? It's just going to get hotter. Not the case. So this is a picture of a chap who doesn't look too happy wading through a flood. This was a picture taken from the northeast of England, just within the last few years as well. And the thing is, in particular, where we live in the British Isles, we're going to face a lot more erosion events, being an island nation. We've also got lots of flooding events as well. So the important thing to remember about climate change is it's going to affect different areas of the globe differently. So it's not just the same warming effect or anything like that. What's important to remember, though, with flooding, obviously, you've got your first effects, which are things like it's harder to get supplies out to people. There's a lot of physical damage. There's lots of emotional damage that comes with floods, if any of you are unfortunate enough to know about that. But there's also secondary issues that come with it, like such as disease. So what happens with floods in particular is that the sewage comes up and it spreads into people's water supplies and things like that a lot more. So you've got to remember, we need to be able to predict what these secondary effects are going to be like too. So as I was saying before, you're probably thinking, well, then why don't you work on the present climate change? Why don't you work on the future? Why are you looking in the past? That's just silly. So the thing is, going back to that principle of uniformitarianism, there's a lot of similarities between the past environments I look at and the ones that are going to happen in the future. So in particular, you can see on this first graph here, we have the Greenland ice sheet and the mass of it declining. Quite rapidly, you can see that too. And what's important to remember is that back in the time period which I study, it provides context for an Earth system with only the developing Antarctic ice sheet as well. So, and already we're losing the velocities of the mass of the West Antarctic Peninsula are speeding up. We're losing a lot of the Greenland ice sheet, which is expected to totally ablate by the end of the 21st century. So already you can see a lot of parallelisms there, which are quite concerning. And on the right here, you can see we have the CO2 concentrations as well. So about 15 million years ago, we had an event called the Middle Miocene climatic optimum. And this was a much warmer, wetter time than we have today. But what was really interesting, scientists found, is that the global CO2 concentrations were really similar to today. So back then, when it was much warmer, wetter, it was about 400 to 600 parts per million. If you look up there, you can see we've already exceeded that in like the last year, or April, just gone as well. And what's really important to remember is that during this time, we know by looking at fossil records and things like that, there was a lot of displacement, species went extinct, there was mass movement between continents, and we need to know how we're going to be able to cope with that as humans on this globe that we've sort of catered to our own needs. We don't know how we're going to actually cope with that in the future. So you're probably thinking, well, thanks for the doom and gloom, but what can scientists do about it? So what scientists do, we can write these reports, not me personally, I'm not that much of an expert, but they write IPCC reports that you can see here. And basically, IPCC reports are a summary of all the recent studies done by climate change scientists and things like that. And the report comes out every few years. It's written by experts in the field. So there could be about 100, if not more, authors on these papers as well. And what they do is by looking at all these studies and collating them, doing lots of things like that, they can say to policymakers and people who change law, like, look, if you don't change the emissions and everything that's going on, this is going to be the outcome. Those emission scenarios, this is some examples of them here. So that's what SSP stands for, basically. It's just the emission scenarios. But on the left here, leftmost, you can see, now this is a scenario where, as a globe, we do start curbing back our emissions. We're very particular about how we're using fossil fuels, things like that. So this is sort of the best scenario that the IPCC stated. So basically, you can see here, so this graph is basically showing the change in global surface mean temperatures, the difference between that of 1850 to 1900 to that of the end of the 21st century, the way that we're modelling things as well. So you can see here that the error bar goes just below 2 for that first scenario there. To put it in context, scientists are worried about 1.5 degrees increase, never mind 2 degrees increase. But we go over to the other side here, we have the SSP 5 to 8.5 scenario here. This is business as usual. This is if we don't change our habits or anything like that. This is not us getting worse. This is us just saying, doesn't matter. And you can see, what's really interesting is that even though the values are getting bigger, and we don't know the absolute outcome of what that's going to be, it's getting more and more unpredictable, hence the larger error bars. So if we really want to know how we're going to mitigate the effects of climate change and things like that, we need to minimize those error bars to actually know how we're going to combat that. And this is why it's really important that we don't forget events such as the COP26 climate change, which happened only in November last year. So it was fantastic to have people like Greta Thunberg, she was outside marching away, saying, pay attention, this climate crisis is going to affect all of us in the very near future. And again, it's really important that we do have people, such as politicians, such as scientists, all coming together so they can actually talk to each other and, try and find these solutions. So it's really important that we don't forget that, even though the world is a very, very busy place. So thank you very much for listening to my presentation. It's been wonderful talking to you. If you do have any questions, please just ask me at the end. So I hope I've given you a little bit more context about how we can look at tiny little dead things and how they can tell us a little bit more about the future. So thank you very much.
Is this what I use? That's the one. Does this work?
That's all good.
Thank you. Oh, that's me. Hello everyone, I'm Chloe Grant and I'm a PhD student here at Newcastle University. My research is looking into a special type of inflammatory cell in the brain of chickens who have broken keel bones. So, the immune system. In mammals, we know that it is inflammation which causes the reduction of neurons in humans, but that is not yet certain in chickens, and that's what my research looks at. The specific type of neuron reduction we're looking at today comes from hens with broken keel bones. So what is the keel bone? That is this long blue bone which runs down the centre of hens. When hens are roaming around the environment, so that is, for example, a free-range environment, those hens can wander around and actually collide with things like their perches and their feeding and drinkers. When they collide with them perches, they can break this keel bone. When that keel bone is broken, that often leads to what I like to call the unhappy chicken. When the chicken is unhappy, that causes the chicken to be very chronically stressed. So you might be wondering, the chronically stressed chicken, why does that matter to us? Well, we want our hens to be as happy as possible. And I think that for anyone who's concerned with animal welfare, for all our animals, we want them to live as comfortable and as happy as a life as possible. But actually, when our hens are unhappy, and which leads to chronic stress, that actually has a knock-on effect for our food security. So when our hens have, for example, a broken keel bone, and they become unhappy and stressed, this actually means that the hen is likely to lay less eggs. So that has a huge knock-on effect for the egg industry. So when the hen has a broken keel bone, which leads to the hen being unhappy, that is caused by chronic stress. Now, in the title, I mentioned a cell called a neuron. It is neurons, and I can't reach, but you can see up there, that long spidery-looking cell is a neuron. And those neurons are what regulate all of our processes in our body. But in this case, it's a specific type of neuron in the hippocampus, which, again, I can't reach. But if you can see where at the top right-hand corner of the brain, the chronic stress caused from the broken keel bone leads to the reduction of these neurons in the hippocampus of the hen, which helps regulate the hen's mood, and that's essentially what leads to the unhappy chicken. So we don't know what causes the neuron reduction. From humans, as I mentioned, the cause is inflammatory. So that's where my work comes in. If it happens in humans where inflammation causes this reduction of neurons, which leads to a poor mood, is it likely to be inflammatory in the hens? So that's where my work comes in, and I'm looking at a specific type of inflammatory cell in the brain called microglia. And I have an illustration of the microglia here. Looks similar to a neuron, a spidery-looking cell. And those microglia are found in the brain, and they respond to inflammatory cytokines, which essentially tell those microglia to destroy the neurons. And when the neurons are destroyed, the bird will have less neurons, and that leads to our unhappy chicken. So I'm looking at those inflammatory cytokines in the brain of chickens, and I've picked two inflammatory cytokines here, which is called IL-1 beta, which stands for interleukin, and IL-1/6. And those two inflammatory cytokines, basically, from the human research, tell the microglia to destroy the neuron. Therefore, we have less neurons and a more general unhappy mood. So, to do this, I looked at two different types of birds. I looked at birds with more severe keelbone fractures, so a worse broken keelbone, and birds with less, more minimal broken keel bones. In the birds with the more severe broken keel bones, they had a greater reduction of neurons, and those birds were more unhappy. So I would expect the birds, so this is the bars in the blue, with the severe broken keel bones, which were more unhappy, I would expect them to be expressing greater levels of IL-1 beta and IL-6. And that would help me give an insight into, is it the immune system causing this unhappy mood and this reduction of neurons in the brain? Actually, there was no difference there. So this tells us that currently it's uncertain if the immune system is causing this reduction of neurons and unhappy mood in the hens like it does similar in chickens. So then I looked at an anti-inflammatory cytokine called IL-10, which basically, from the human literature, we know tells the microglia to stop producing inflammation and to stop driving that neuron reduction. I would have expected the birds with the less severe keelbone fractures, who were more happy than the birds with severe keelbone fractures, that they would have more IL-10, because I would not expect there to be the presence of inflammation. Actually, I found the opposite. I found that the birds with the severe keelbone fractures, the birds which were more unhappy, the birds with the greater neuron loss, had more of this IL-10. So this is the opposite of my hypothesis, where I would have expected going back these cytokines, the inflammatory ones, I would have expected it to be the reverse, but actually it was this anti-inflammatory cytokine where the birds, which were more unhappy, had more of. This tells me that there could have previously been inflammation, and this is the brain's way of saying we've had stress from our injury, we've lost a greater level of neurons, we're unhappy. Now it is time to actually go back to normal inflammation levels, and for those microglia, those inflammatory cells, to return to a normal state. So next, I thought, well, I'm going to correlate this with the level of neuron loss. So in the blue, again, on the upwards column, this is our neurons. So as you can see, it starts off the blue one relatively high. We see a negative correlation where the greater the neuron loss, the more IL-10 those birds were expressing. So this again backs up my thoughts that there has previously been an episode of inflammation, and that IL-10 is being expressed to tell the microglia cells in the brain, stop destroying neurons, stop driving this inflammation loss, and let's return back to a normal, happy, healthy brain. So now, this leaves me in a bit of a dilemma. Is it the immune system? It could be the immune system. We don't know. So next, I'm going to look at those microglia cells in a little bit more detail and try and find out a little bit about the character of them. So this is a tiny little microglia here. That's what it looks like under the microscope. I'm going to do the same again take the birds with the worse keelbone damage, the birds which were more unhappy and had greater neuron loss, and compare them with the birds who had less severe keelbone damage, who were in more of a happy mood. I would expect the birds with the greater neuron loss, the more severe keelbone fractures, to have more of these microglia cells. To do that, I'm going to be looking under the microscope and using a specific marker which only microglia express to be able to actually count those cells. So this work is currently, if I can get the antibodies to work, I'm going to be counting them and looking at the general shape of them. And the shape will be able to tell us if the cell is producing inflammation, because it will actually look a little bit less spidery and more swollen. So that is work that is currently going on, which is the next chapter of this work, to really investigate, is the immune system the driver of the neuron reduction in birds with broken keel bones, which leads to the unhappy chicken. So, thank you for listening. So today we've discussed that inflammation is at the cause. We're not too sure yet. And we actually have increased IL-10, which suggests that inflammation could have been present, but we're not yet sure. So thank you for listening.
Just testing. Yeah, brilliant. So, good evening. We are Team Sugar, and it comprises of myself, Corin, Alex, Tom, Anna Chiara, and also in the audience there, Kitty. And we're going to discuss with you today our project, which is in collaboration with the European Space Agency with their Fly Your Thesis 2022 program. So to give you a little bit of an insight before we get into further detail, we're going to be looking at this. It looks quite blurry, as you can imagine, on a big screen, but this is a yeast cell. And we're going to see, if we give a yeast cell glucose, how much will it uptake? But we don't want to do that on the ground, no. We want to look at it in abnormal conditions. To do that, we need to go on a parabolic flight. So later this year, in Bordeaux, France, we'll be on board the Airbus 310, run by Nova Space. Previously was Angela Merkel's answer to Air Force One. 2015, she realized she didn't really need it, and now it's used for scientific experiments and public experiences. So, as the name suggests, parabolic flight is you're in a plane and you go in a parabola up and down many times. But there's 3 phases to that. You have the nose-up phase, where you experience hypergravity, so that's 1.8 G, so you effectively feel double your weight. Then you have the arc, the trough right there in the middle, where you effectively feel weightlessness. It's microgravity, nearly 0 G. And in the end, you have the nose down point, where you again feel hypergravity. Now, unlike your normal planes that you may get on to go on your holidays, you in fact have 3 pilots on board these flights. You have one who controls the pitch, that's effectively the nose up and down. You have a second whose sole job is to control the wings, the side to side. And then you have a third pilot whose job is to do all the standard stuff you would expect on a plane. The cabin pressure, the engine speed, and also the temperature. But all of these 3 work together to ensure that we experience exactly the G that we expect at each moment in time. To ensure there's no turbulence, we don't do any of these flights on land. Well, obviously you do them in the air, but you know what I mean. You actually do them over the sea in the Atlantic Ocean, just off the west coast of France.
Okay, so you are probably wondering why the name of the team is SUGAR. SUGAR is standing for Saccharomyces cerevisiae, uptake of glucose applying real-time imaging. The Saccharomyces are a budding yeast or baker yeast, which is the common type of yeast that you can imagine for making the bread or the beer. But, well, apparently is not only used for this amazing purpose, because it has been widely used as a model organism for studying some processes in cells, for example, some cell signaling processes. So have been widely used for scientific experiments, mainly for 3 reasons, because the yeast cells are very robust organisms. It's very easy to grow yeast cells and to transport yeast cells if we compare with mammalian cells. And even they had their entire genome mapped in the 1996. The uptake of glucose is a cell signaling process by which the cells can sense and can respond to the signals from the surrounding environment. And you can see here, this is a yeast cell, as Corinne showed before, very bad in this picture. And this is a fluorescence image of a yeast cell. So you can see in green the fluorescence, because what you can do and what was done is to make the glucose fluorescence and to observe how the cell can uptake the glucose. And actually, you can do it in real time. So our idea is to do it in real time during the parabolic flight. And for doing that, we are building up a microscope, a fluorescence microscope, that, of course, shouldn't be like just a normal microscope, because this microscope should be able to withstand abnormal gravity conditions. So it should withstand vibrations. So, given just this information, the aim of our project will be to image fluorescent glucose, which will be uptaken by yeast cells during a parabolic flight in abnormal gravity condition, meaning hypergravity and microgravity condition, and to compare the rate of the glucose uptake by these cells with the cells that will be grown on the ground and will just, experience what we are experiencing every day, just, normal ground life. And what we will be doing is we will take some images in real time during the flight, some video more than images, and this video will be post-processed after the flight, and we will see, in this way, the different rate of the glucose uptake. The analysis will be done with some software that were created in the Hullman lab in medical school. So, which is the rationale, or I would say, which are the hypotheses behind our experiment? So, the cell signaling is a process that has been demonstrated to be impaired by abnormal gravity condition. And the real-time microscopy can enable us to direct observe in real time what is happening. So during the flight, from the moment we are giving the glucose to the cells until the end of the flight. And this is the first time that something like that was done on a parabolic flight, of course. And well, the third hypothesis, as I mentioned before, the yeast is a very robust organism and is a very good model for studying cell signaling process.
The flight campaign is in October and lasts for 2 weeks. Some onboard rules are no free floating while we're in zero gravity. We must stay within our own section of the aircraft because there's other teams on the aircraft performing their own experiments. And we get 2 team members per flight to go up. Moving on to the design, the yeast cells are encased in these microfluidic channels, as shown in the center figure. And there is 30 of these, 1 for each parabola. A syringe pump injects fluorescent glucose into the microfluidic channels, and any squeeze out is collected in IV bags. A fresh channel is moved above the microscope between parabolars by a motorized stage, as seen on the top right. And the microscope itself will take both bright field images, so like a standard image that you see in a microscope, and a fluorescent image. And the variables we're going to be looking at are the time of delivery of the fluorescent glucose at different points in the parabola, and the concentration of fluorescent glucose. So we have to overcome a few design challenges. First being, everything needs to be secured to the aircraft at all times. And second being, we're dealing with fluids, so the whole experiment must be contained and autonomously controlled. The rig itself is comprised of 2 parts, a control base and an experimental base, both of which are bolted to the seat rails of the aircraft, so they don't fly about and hit anyone in zero gravity. The experimental rig, which contains the microscope, is on dampeners to reduce vibrations coming from the aircraft that may affect the image quality of the microscope. And the experimental section is contained inside a waterproof Zargis box, and the microscope, the stage, and the syringe pumps are controlled from the outside of the box on the control panel.
Okay, so the scientific value from this experiment comes from two places. It comes from the novelty. So this kind of experiment has never actually been done before, looking at real time. So it's been done before, so samples have been sent to the International Space Station and they've been imaged before and after, but during the actual microgravity phase, they were not imaged. So this is for this experiment, it's the real time that's the novelty of this. The second thing is the applications for the future, and this can be split up into three sections. You've got the astronaut health, synthetic biology for food and medicine production in space, and the third thing is stress testing the microscope components that we're going to be using. So just to talk about those three things here. So the first thing is astronaut health. So because yeast is a model organism, the results that we get can be generalized to form a hypothesis as to what would happen in a mammalian or human cell in microgravity. So this is going to be really handy information to know for long-term space travel. The second thing is synthetic biology. So a synthetic cell is a cell that has been modified to produce chemicals and food and medicine for us to use. And this is done by modifying genetic material inside the yeast cell, shown in figure B here, and then also in figure A, this synthetic bacterial genome is then transplanted into a bacteria cell by genome transplantation, and this is what forms the synthetic cell. So this has applications in space travel, because then you can make like food and medicine in places where you wouldn't have a lab to do so. The last thing is future missions. So again, our microscope uses components that haven't been tested in these kind of conditions before. So our results that we get can be then used for future missions. So when people need to image anything in a microscope in zero gravity, they can look at our experiments and see what worked and what maybe didn't work.
So just to give you a brief summary, we're going to be taking yeast cells up on a parabolic flight and imaging them to see how they uptake glucose during these abnormal gravity conditions. And as Tom's just explained, there's so many applications that this can be used for. But another part of our work is sort of showing people how their current research could actually maybe be applicable to spaceflight and space research. So it's just something extra to think about. But to be honest, what are we really excited about? Well, the experiment's all well and good, but we actually are going on a zero-G flight, which we're all extremely excited to be able to feel near weightlessness. Less excited to feel double our weight than we do on the ground, but you know. And these flights normally cost around 10,000 euros per participant. So for a group of students to be able to go up and perform an experiment, we're pretty excited. So thank you so much for coming here this evening, and thank you for listening.
Yeah, thank you.
We should give an extra big round of applause, actually, because we didn't manage to do applause in between the talks. So to all of the speakers, thank you very, very much for your talks tonight.